So good morning, everyone. Thanks to those who actually made it here. Thanks, of course, also to the speakers. I think we start nevertheless because the schedule is so tight. So I'm sure there will be people dropping in as the session progresses. So welcome to our session on parasites, drug resistance, the potential impact of nanotechnology. And the session is started by two local speakers, and we get more and more global as the session progresses. And the session is opened by Professor Reto Brun of the Swiss TPH. Reto is one of the motors that have been driving drug discovery for protozoan parasites, and nobody is better suited to open that session than him. Thank you very much, Reto.
Thank you, Pascal. I thought I'd give you a short introduction into neglected tropical diseases, parasitic diseases, drug-resistant parasites, and the nanoparts. They will probably come with the later presentations. So, the World Health Organization defined approximately 20 diseases as neglected tropical diseases. These are diseases which affect the poor populations in poor countries, and they add to poverty, which is a catastrophe. So these neglected diseases, they are comprised by diseases caused by protozoan parasites, by a list of helminth diseases or worm diseases, by a few bacterial diseases, and by viral diseases. The protozoan diseases and the helminths, we group them as the parasites. These are the parasites. And they affect approximately 1 billion people on this planet. And they have a great impact in that they add mortality and morbidity, as you can see. Control of these diseases mainly depends on drugs. And during the past 40 to 50 years, not many new drugs were developed for these diseases. So the existing drugs, they have massive limitations. And one of these limitations are drug-resistant parasites. And drug-resistant parasites are responsible that we constantly have to develop new drugs to overcome the loss of the ones we lose to drug resistance. During the past 20 years, many new initiatives were founded, so-called public-private partnerships or product development partnerships. The goal is to come up with new products and to close the gap between drug discovery and drug development. So you see here some of them, and for us, and you will hear more about the medicines for malaria venture and the drugs for neglected diseases initiative. First successes of these initiatives can be reported. I'm very happy to do so. So I can present two potentially new drugs for sleeping sickness. Sleeping sickness is one of the most neglected diseases, restricted to Africa. And the organization which is very much involved here is the Drugs for Neglected Diseases Initiative. And they have there is one molecule in the pipeline which might come out, enter the market next year. And this is called fexinidazole, and it would be the first oral drug for sleeping sickness. There is a second molecule in the pipeline which is even more promising. It is also a new chemical entity. It's a benzoxaborole, and based on its favorable pharmacokinetics, it can be expected that a single oral dose will do the job. And this is just fantastic. Now, when we look at malaria, and here we constantly need new drugs because drug resistance is really a very, very severe impediment here. We also have new products, and the major player in the malaria drug business is the Medicines for Malaria Venture Initiative. And this molecule OZ439 that's a long story. I was personally involved in this. It's a synthetic peroxide which should be able to replace the artemisinins. And you see two combinations, they already passed two phase 2 clinical trials. Another molecule, this is also representing a new chemical class, and this is important in terms of drug resistance development. This is a molecule from Novartis. It is a spiroindolone, and it is also already in phase 2 clinical trials. Now, drug resistance, we know it, of course, very well from bacteria. There it's extremely prominent, but we also know it well from malaria, where virtually all the new drugs sooner or later become resistant. And this is a bit sad, because with the artemisinins, there was hope that they could sort of be the exception, but resistance development already starts. Now with African trypanosomes, and I want to give you this example. The picture is a bit different. We have massive resistance among animal pathogenic trypanosomes, but among the human pathogenic ones, those are a bit different parasites, but very much related. We hardly see any resistant strains. Well, the explanation for this is rather straightforward. In animal pathogenic trypanosomes, we have very low numbers of parasites per patient. The infection rates are low. The transmission is extremely poor compared to the animal pathogenic ones. And underdosing, mass treatment, and prophylactic treatment, as we know it from the animal side, is not being used in humans. So actually, the drivers are simply not there. If they would be there, I'm sure we also would see much more resistance. Drug resistance is also a big problem in helminths. Virtually all the drugs, they sooner or later become resistant to the new drugs. And helminth diseases, you know, they are equally distributed to humans and to animals. And here again, it's interesting to see that among the animals, there is much more drug resistance. But again, it has to do with the use of the drugs. And here, in animals, the treatment frequency, underdosing, mass treatment, and also single drug schedule. These are drivers of this resistance. Now, 5 years ago, the London Declaration was signed by major pharma companies, by WHO, by donor agencies, by affected countries. And the goal is to eradicate one neglected disease, that's the Guinea worm disease, to eliminate some and to bring others under control by the year 2020. Now, sleeping sickness is on the radar for elimination, and thanks to massive control measures, as you can see, the number of reported cases and of estimated cases came down tremendously during the last 20 years. So elimination is defined as bringing the number of reported cases below 2,000. By the year 2020 is a realistic goal. But colleagues of us at our institute, they looked at the estimated costs of this whole enterprise. What would this elimination cost? And you can see the numbers are enormous. So in the best case, it would be about $1 billion. And also the time frame is long, because it's not only until you reach this situation, but you also have to have a 10 to 15 year follow-up period. So it's a costly business. The first disease which probably can be eradicated is the guinea worm disease. And this is a debilitating and very painful disease. And the numbers are impressive. 30 years ago, we had 3.5 million cases. And in the year 2015, there were just 22 cases. This is all thanks to the Carter Foundation and also to WHO. But here recently, I read an article saying or stating that guinea worms were found in dogs in Chad, and this could mean that dogs could be a reservoir. And of course, this could complicate the story quite a bit. The elimination of neglected tropical diseases by the year 2030 seems to be realistic. What is needed is political will. The political will is there. We need significant funding. I dare to say also funding is there. And we need better tools, especially better drugs. But then we have the resistant parasites, and they force us to come up with new drugs all the time on a consistent basis. And then it's important that we have a strategy to protect these new drugs. They cannot just be used like that, but they have to be used on a restricted basis, and they have to be used in combination. This helps tremendously to slow down resistance development. And with this, I want to stop, and I thank you very much for your attention.
Thank you very much. Early morning questions.
What is the main obstacle or the main trigger of resistance evolution? Is patient compliance, or that we do not find sufficiently good drugs, or maybe the way we are administering those drugs?
It's not the problem of the drugs. I believe that there is no drug which is completely resistant to resistance development. It is just the use. And the more we use the drug, the faster it goes. And also the number of parasites is, I think is very essential. You see, in malaria, in a heavily infected patient, you may have up to 10 to the 11 parasites. In a sleeping sickness patient, maybe you only have 10 to the 7. And then the drug use, of course, is very important. Yeah, compliance, underdosing, all these things. But I'm afraid to say that once the parasite number is large enough and the drugs are freely available, then there is a danger, and sooner or later resistant parasites will show up.
Thank you very much.
Okay.